Now, disease is due to alteration in the imprinted region. I'll make you understand this. See, it has been known, you know, that there is one pair of chromosomes, isn't it? One is coming from mother and the other is coming from father. It was thought that it is an actual pair. There is no difference between both of them. But now it is understood that certain chromosomes, not all, has got some functional differences. Okay, depending upon from which father it is coming from. And this functional differences is due to a process called genomic imprinting. Okay, it's due to a process called genomic imprinting. Now, what is genomic imprinting? That is silencing or inactivation. Okay, inactivation or silencing of certain genes in certain chromosomes due to epigenetic process. Inactivation of certain genes during gametogenesis, that's important, of any parent, mother or father. Okay, during the gamete formation, some genes are silenced. Maybe some genes here are silenced, some genes here are silenced. It's the same chromosome. So if it is going to the child, both are not going to be the same, isn't it? So two different parents I'm talking about. Okay, so this is from father and this is from mother. If it is going to the child, it is two different things. Though it is a pair, it's two different chromosomes. Okay, so there's a functional difference due to genomic imprinting. Genomic imprinting means it's an inactivation of certain genes in chromosomes. Now we are talking about diseases of genes, not chromosomes. Inactivation during gametogenesis. Okay, which is an epigenetic process. Why is it called it's an epigenetic process? Why do they say it's an epigenetic process? Because usually alteration is due to what? Change in nucleotide sequence and all those. Okay, CGG becomes CAG, like that. Okay, we'll study this in detail in biochemistry. What is the nucleotide sequencing and all those. But epigenetic means, that is, there is no change in the nucleotide sequence. There is something known as methylation. One example of epigenetic process is methylation. So it's epigenetic. Okay, epigenetic factor. So there is methylation of cytosine residues. Okay, there is methylation, just note it down, methylation of cytosine residues on the promoter region. All these, it's need lots of explanations. Don't worry, we'll understand DNA replication and all those. Okay, so at one end you have got this DNA. At one end there is, for example, 5'3' end. So what happens is that methylation takes place. Okay, methyl group just gets attached to a cytosine residue there. Okay, you know that there will be cytosine. So methyl group comes in attached and this inactivates that particular gene. So that's the epigenetic process. Okay, and that process is known as genomic imprinting. Okay, inactivation of certain genes during gametogenesis, which is due to epigenetic process, which is methylation of cytosine residues in the promoter region. Now, as you could see, this can be either paternal genomic imprinting or paternal imprinting. It can be a maternal imprinting. That is, what does it mean? Silencing of a region in father's chromosome, or it can be silencing of a region in mother's chromosome, one or two regions. Okay, so it's called paternal imprinting and maternal imprinting. So the disease is seen in both. Same chromosome. One region is inactivated and it has been transferred, causing a disease. This region is inactivated, it's causing a different disease. Same chromosome. So that's a functional difference within the same sets of chromosomes. Not different, same. Okay? So hope you're getting this. What is it? It's a functional difference exists between the two pairs of chromosome which was received from father and mother, which is due to genomic imprinting. What is genomic imprinting? Inactivation. And one of the process of inactivation is methylation of cytosine residues. And if there is paternal imprinting, there is one disease. If there is a maternal imprinting, there is going to be a different disease. So let us see what are the diseases. Now, as a continuation of previous one, I said it can be either paternal or maternal. Maternal means mother, paternal means father. Okay, now if the same chromosome number 15, okay, the chromosome number 15, the Q arm, region 1, band 2, now you know it. Okay, so 15th chromosome, Q12, if this genomic imprinting takes place, the paternal chromosome, you get something known as Prader-Willi syndrome. Okay, Prader-Willi, W-I-L-L-I. So it's Prader-Willi syndrome. Now the same thing in the same chromosome, in the same region, happens in maternal chromosome. It's not the Prader-Willi syndrome. It's called Angelman syndrome. Okay, Angelman syndrome. Okay, now don't have doubt your mother is man. Okay, mother is man. It's much more easy. Paternal Prader-Willi. So learn one, the other is the other way around. Okay, so it's the silencing of a particular region of a chromosome of father. So same chromosome, but the effect is different. That's what is atypical inheritance. Normally, if it is Mendelian, it should have been the same disease, isn't it? Because that is, it should have been the same manifestation. But we have two different manifestations.